Okay, so this chapter largely deals with human interactions with other species that cause disease-specific outcomes. So we're going to start talking about a little bit of ecological concepts, and then we're going to talk about macrofauna. But honestly, macrofauna is probably a pretty small part of this, and then we're going to focus mostly on microbes and microorganisms. But yeah. So if you consider an animal with any specific factor, you can assume that they have a bell-shaped distribution of things they can tolerate. So for instance, temperature in lizards, they tend to be fine in a tropical environment. They can survive up to like Virginia, specifically speaking of green anoles. But if they get too far north, it's too cold and they die off. And so their temperature tolerance would basically be a bell-shaped distribution. They have an optimal area, and then they have areas that they can tolerate, but they don't do as well. And then eventually there's areas that they can tolerate if there's zero competition, but under no other circumstance. And so if you take that range of tolerance and you plot a different range of tolerance on the y-axis, say for water resources or something like that, there would be an intersection of those two bell-shaped curves, right? And that intersection would be the range of tolerable habitats for those two factors. If you do a third one, you end up with a three-dimensional sphere, right? On the z-axis coming out, you would end up with a circle, right? Which is hard to visualize and hard to draw, so I'm not really going to try to do it. But basically, that's what you end up with. So this image is kind of trying to show that, but instead of using bell-shaped curves, it's using tolerable ranges. And you can see if you take food size, branch density, and temperature, which are three factors you end up with what's known as a, well, it's a square. It's a three-dimensional shape. And if you take all of the factors of an environment, say there's 462 factors of an environment, right? And you were to graph all those things, you would end up with a 462 dimensional object, which is impossible for humans to visualize because we can't think of anything more than 3 dimensions. But that's a hypothetical that exists. That is the concept of a niche. So you can just think of it in 3 dimensions if that makes your life easier. But you can imagine that 3-dimensional space that you're seeing right there is all the areas that that animal can occupy. Okay, this is what's known as a fundamental niche. So then the question is posed, what if two niches overlap? And if two niches overlap, what you end up with is competition. And in competition, basically what happens is one organism is probably, dog's gonna start drinking water. One organism is probably gonna have to reduce the size of their niche slightly. And so the easiest example is, or the classic example from the textbook is the bluegill and the green sunfish. They both occupy similar niches in that they both like to live in small ponds, lakes, and rivers close to the shore, and they like to eat little insects. The green sunfish is more aggressive than the bluegill. So when they're in the same niche, what happens is the green sunfish will chase the bluegill out of their area. But the bluegill have an important adaptation that the green sunfish doesn't. Namely, they can occupy littoral waters and basically filter feed for small microinvertebrates, and they can make a living doing that. So what they're doing is they're expanding their niche. And basically what ends up happening is if the fundamental niche is all the areas that can be occupied, the realized niche is what they tend to occupy, and it's almost always smaller than the fundamental niche. That's known as character displacement. There's a concept known as competitive exclusion principle. Two organisms cannot occupy the same niche in the same location because there will be competition. One of them will win, one of them will lose. So organisms work to reduce competition amongst conspecifics. So that's a competitive interaction. You also have non-competitive interactions. So for instance, predation is a competitive interaction. Predation is a positive event for one of the two organisms and a negative event for the other organism. So in the zebra and the lion, it's positive for the lion, it is negative for the zebra. And it's marked by a relatively short interaction and a rapid death. And I mean relatively short interaction because it can be measured in hours at the longest, and the death is no longer than hours, as opposed to parasites and parasitoids, which can go over the course of months. Parasites, yeah, so parasites. That is a negative for the host and a positive for the parasite. It doesn't always end in death. Some parasites don't kill their host. So the organism on the left that's disgusting, I can't remember what they're called, but they're a type of water bug. And basically what they do is they swim up the fish's gills and then they eat the fish's tongue off. And then they kind of attach themselves to the host's tongue and they begin to serve as the host's tongue. And they actually connect their blood supply to the host, which is really, really weird. But that's an example, a pretty extreme example of parasitism. You can also think of like ticks and fleas and stuff like that. There's lots of parasites. Parasitoids are a little bit different. That's a negative interaction for the host, a positive for the parasite, but it's a long, slow, painful death. A lot of wasp species do this, wherein they inject their eggs into a host, and then the eggs eat the host from the inside out. So that's what I mean by long, slow, terrible death. Mutualism is beneficial for both organisms. So the ant in the acacia tree is a mutualistic interaction, wherein the ant provides defense for the acacia tree, and the acacia tree provides nutrients for the ant. The fungus have a lot of mutualistic relationships with trees as well. Humans have mutualistic relationships with lots of organisms, but we'll talk about that later. There's also commensalism. That is basically a neutral interaction for the host and a positive interaction for the parasite. Barnacles and the whale are a classic example. The barnacles can annoy the whale, but they really don't do anything to it. The whale basically just doesn't notice that they're there. It'll try to slough them off, but it really doesn't matter much. It's good for the barnacle because that allows it to move around the world, which gives it more opportunity to reproduce in more areas, and it allows it to attract food from everywhere. But the whale basically doesn't even notice. They're just kind of like, that's part of life, is that you have barnacles that grow on them, right? You can think of the same way of like the little animals that live in your skin, or some of the organisms that live on humans as well, mites and stuff like that. We basically don't notice that they're there, but it's good for them. Spite is something that's almost always left out of textbooks because it's really not a biological phenomenon, it's a human phenomenon. Basically, it's going out of your way to mess up someone else's day. So it's expending energy and effort to cause harm to someone else, right? And it's basically only found in humans, and they were able to replicate it in primates in one very specific study. It's interesting because if you go to the Wikipedia page for spite, there will be a huge list of spiteful behaviors like infanticide in lions and stuff like that, but they're not truly spiteful because they are actually causing benefit for the actor. To be spiteful, it has to cause no benefit for the actor and no benefit for the recipient of the behavior. So coevolution is basically when two species evolve in response to each other. It could be a mutualistic relationship, but oftentimes it's an antagonistic relationship, and that tends to be the evolutionary arms race. So, you know, the crab eats the periwinkle, the snail. So the snail develops defenses. The toughest shells are the ones that survive. So then the crabs have to get stronger pinchers to break the shells. So then the snails have to have stronger shells. Only the stronger shells survive, and so forth, back and forth, back and forth, back and forth, until you have heavily armored prey and predators with extremely powerful defenses. But it can also be mutualistic, right? So the acacia tree and the ant are obviously an example of a mutualistic coevolutionary relationship. So we're not going to talk Well, we're going to talk about antagonistic ones, but it's mostly going to be endoparasites. It's not going to be ectoparasites. Because to be honest, there's not a lot of antagonistic evolutionary arms race going on with humans. We don't have any predators. So the book kind of glosses over evolution with large mammals, and so I would spend a little bit more time talking about it. Because there's been a lot of animals that we've had evolutionary relationships with that are very beneficial for us. So one of the first is dogs. Dogs in this day and age are largely used to provide comfort and recreation. Like my dogs don't do anything for me besides make me happy, and I'm okay with that. I mean, I spend a ridiculous amount of money on them a year to keep them happy, so I mean, it's working out, I guess. But broadly, dogs used to be functional for humans. They help protect our sheep and our flocks. They helped us hunt. And they still do a lot of those functions, but most of them are just for enjoyment. Cats are interesting because they actually domesticated themselves. We didn't like capture cats and say, hey, why don't you be a cat in my barn? The cats saw the mouse problem and they just decided to go in and take care of it for us. We thought this was good, so we started giving them milk and cream, whatever, feeding them because they would come around and kill the rats. And it largely helped us with our grain storage problem. Been a great situation. The interesting thing about cats is they really still fulfill that function. If you go to any farm around southeast Minnesota, it's basically a necessity to have barn cats. They help keep the rats under control. And so that is still a function and a viable job that they do. They've also become pets where they are spoiled and treated like the princesses that they think they are. Cats are really interesting. It's also really common, apparently every state except for Minnesota allows cats in the brewery because the cats help take care of the mice problem. Breweries have a lot of grain, so they have a tendency to get mice. The nice thing about breweries is that they boil the grain before they use it, so the grain is sterilized no matter what it's exposed to. But it is still a problem, like the loss of grain. If you find a mouse in your grain, you have to throw it out, right? But a lot of breweries will actually keep cats. But apparently Minnesota doesn't do that. I personally would very much enjoy brewery cats. Cattle broadly have been domesticated with animals, been domesticated by humans, and this has caused a lot of effects that we've talked about before. Basically, a steady supply of meat. It's caused changes to the cattle themselves. As you can see, the aurochs were the species of the predecessor to cows, more or less, and they were much larger and they're more aggressive. We bred them to be more docile, a little bit smaller, easier to manage, and cows have probably been one of our most closely related species over the generation. This isn't one you typically think of, but like mice and rats. Mice have co-evolved with us, much more of an antagonistic relationship than a cooperative relationship, but they basically followed us everywhere we've gone, either intentionally or unintentionally. We've introduced rats to basically every place that we have visited. So that's been a coevolutionary relationship as well, but not really one that would be mutualistic per se. So coevolution with microorganisms is very, very challenging. The first of which is that it's estimated that there's 100 million species. Just the sheer number of microorganisms in and of itself creates a huge problem. The 100 million is estimated. I mean, and those estimates are not great, right? There's a lot of organisms out there and we don't really know how many there are. And the diversity of organisms can also be challenging because, I mean, you have bacteria and viruses, fungi, protozoa, which is actually not really a group, but basically eukaryotic single-celled organisms, and then algae are all different microscopic organisms. And because they have different cellular features and they're not very closely related to each other evolutionarily, something that works on a bacteria may not work on a virus, and something that works on a virus may not work on a fungi or a protozoa. And some of them have cellular machinery that's very similar to our own. So like antifungals in general could possibly interact with human cells. So finding drugs to treat funguses can be very, very challenging. Another challenge of microorganisms is that they evolve incredibly rapidly, right? Um, Richard Linsky is really famous for doing what's known. He's done this long-term evolution project, where basically, as of like 2014, he's raised like 58,000 generations of yeast, and it's been basically, let them grow 12 hours, put them in a new vial, let them grow, new vial, and he's looked to see what type of evolutionary changes have happened to them overall. And specifically, he's created specific evolutionary pressures to see how they respond to them. And the number of times it takes, like the number of generations it takes for a mutation to occur that's beneficial to a specific challenge. One specifically is like tryptophan, the inability to produce tryptophan. And he's found that they actually evolve a stopgap to that really quickly. The other thing about bacteria is they have something called the SOS response. And basically what the SOS response is, if they detect a stressor, they'll start to repair their DNA, but they do it really sloppy, which actually causes mutations to occur more rapidly. So in the face of a pressure situation, evolution is actually part of their adaptive strategy. They will intentionally try to evolve more rapidly in order to find a solution to the problem. So another challenge that microorganisms pose is that oftentimes they have animal vectors. And the reason that this is challenging is because basically, if we were to eradicate the entirety of a virus from a human host, the virus could still exist in the animal vectors, and we would either need to completely destroy that animal vector or completely destroy the virus in that animal vector. And so that makes it really hard to eliminate some diseases, specifically like bird flu, measles has animal vectors. Rabies isn't a great example. Black Death is one that we really can never get rid of, just because it always has a virus, or like an animal vector to hide in. This also poses a problem because mutations can occur in those animal vectors. So it could be something that's relatively benign in humans, like a COVID virus, right? But if it picks up a mutation that's pathogenic to humans, then all of a sudden it could become a problem. And that's what happened with COVID-19. The coronaviruses largely tend to be not terribly bad. But COVID-19 happened to pick up a mutation that caused it to be lethal, and it caused it to spread very rapidly amongst humans, and so that's why we had the global pandemic. Similar things have happened previously with like MERS and SARS, but they didn't spread as rapidly, and they were able to be isolated before there was a huge problem. So that tends to be something that's very tricky when we're talking about managing viruses. So if you think about the evolution of microorganisms, there's trade-offs. Viruses are making trade-offs just like we're making trade-offs, right? So one of the first things is virulence. Like, how much damage does the virus cause? If it's a very, very virulent disease, it has to spread very, very rapidly, because if it's very violent and causes severe symptoms, that's going to knock the host out and cause them to just stay home, right? But if it doesn't cause a whole lot of damage to the host, then the host will probably walk around spreading it. I'm almost certain that there are viruses out there that cause next to no symptoms, and they spread very rapidly. We just don't care about them because they don't really cause symptoms, right? Like, if they don't hurt anyone, then who cares? Virulence can be associated with the transmission itself. So a good example of this is the cholera bacteria. The cholera bacteria cholera kills more people worldwide than, it's one of the top killers in the world. And cholera has a tendency to break out in these massive massive epidemics of a localized area. And the reason is because it's waterborne. So what happens is somebody ingests the cholera bacteria grows, divides, and reproduces. And when it's at sufficient numbers, it secretes a toxin that causes terrible, terrible diarrhea. That terrible diarrhea helps it spread by getting into local water supply, and then other people drink the contaminated water, and then they get cholera. And so the virulence, the terrible diarrhea that it causes, is actually part of how it spreads. The other thing to think about is how quickly and how easily does it spread. Viruses that spread very, very, very rapidly can afford to be highly virulent. Viruses that do not spread very well have a tendency to not be very virulent. So it's a trade-off between those two things, right? Transmission is largely measured by something known as R0, and R0 is basically if you are infected with it, how many people do you infect with it? And if the R0 is greater than 1, it will proliferate and move through the environment. If it's less than 1, it has a tendency to stay put. And you can see that measles has an R0 of 14. So one person with measles will infect a shit ton of other people. Rhinovirus is a basic cold, and you can see it has a high R0. Smallpox is a very, very deadly virus, and it has a high I0. MERS we were really, really lucky that MERS did not become a pandemic, because that was a highly virulent, it killed a lot of people who got it. And it actually has a higher R0 than COVID-19, and it was basically rapid intervention and sheer luck that caused it to not be spread. Well, as I was kind of alluding to, or directly saying, I can't remember. If a disease is highly virulent, if it causes severe symptoms, it has to be very, very transmissible. Because the host is gonna know that they're sick, and they're gonna feel like crap, and they're gonna stay home. If a disease has low transmission, it needs to have low virulence, right? And so a good example of this is actually STDs. STDs have very low transmission. Like, you have to have sex with the person to pass them from person to person, right? And that's not a very easy way to pass the disease from person to person. So it has to have either long latent periods like HIV. HIV has latent periods that are measured in months to years. Or it has to just not cause a lot of symptoms. And a good example of that is herpes simplex 1. Herpes simplex 1 causes cold sores, right? And you don't always have a cold sore, and even if you don't have a cold sore, you can pass it on to someone else. Cold sores in your mouth, just to be clear, which is not exactly an STD, but whatever, it's close enough. You pass it by kissing, but you can also get it by drinking someone's soda, but it's a lot easier to get by kissing someone. But anyways, that's not something that you do with a lot of people, and so it has to have low symptoms, otherwise it would never get passed on, right? The other thing about herpes is that you have it for life, which means that it can be passed on for a really, really long time. I thought of another example. I can't remember if it's chlamydia or gonorrhea, but it basically has no symptoms in the female. It only shows up in the male. So a female cannot know that she has it because it causes no symptoms whatsoever, and then males become infected with it if they have intercourse with her, which is, it makes a lot of sense from an evolutionary point of view. But anyways, this trade-off has to exist. Thing that tends to shape a virus's virulence is how long it's interacted with humans. The longer diseases interact with humans, the less virulent it is, usually. This isn't always true, though, because mutation is random. So if a random mutation that pops up that increases transmission, then there's a less of a pressure to reduce virulence, right? But increased virulence can also increase transmission sometimes too. So that's not necessarily true, and there's plenty of good examples of viruses becoming less virulent as they've interacted with humans, or There's plenty of examples of viruses that have not become less virulent. Ebola, HIV, dengue fever, yellow fever, Lyme disease, malaria, all of those continue to be highly virulent, even though they are relatively ancient diseases. But there are lots of examples of viruses that become less virulent over time. So like the 1918 flu, the Spanish flu that was going around, still circulates today, but because most of the population has had some exposure to it, it's less of an issue today than it was back in the day. The quote at the bottom was a paper that came out. But basically, the lower severity of Omicron, they argue, is coincidence. And it might be a trend, but it could go in the opposite direction as well. There could be a new strain of COVID-19 that comes out that is very, very highly virulent. So just so that this is an evolutionary trend doesn't mean it's an evolutionary rule. This isn't something that always happens. It's just something that usually happens. But we can use this characteristic to our advantage. And the basic idea is what's known as attenuation. So basically, if you take a virus and you put it in chicken cells and you pass it from chicken to chicken to chicken to chicken, or if you put it in human cells or cow cells or whatever you want to use, and you pass it from one individual to the next and one individual to the next and one individual to the next in a laboratory setting, like in a petri dish, over time, it doesn't need to have any virulence to pass. So you kind of artificially select for variants of the disease that are less virulent, right? And if you get a variant of the disease that basically has no virulence, then you can utilize it as a vaccine. So you can expose people to it knowing that it has no virulence, and that allows it to provide immunity without any kind of side effects. And that's how you make an attenuated virus. That's what's known as attenuated live viruses. The other method is to take the virus and break it up into little bitty pieces, or the mRNA ones where you basically just give a piece of mRNA. Okay, we're going to talk about the human immune system, and the human immune system is really complicated. It's not one of my favorite things to talk about just because it's so complicated and it's like whatever. But something to keep in mind is when we're talking about the human immune system, you kind of need to think of the human body as a metasystem because we have more bacteria cells inside of us than we have human cells. And so your microbiota is an ecosystem in and of itself. And there are interactions between the different various microbiota that interact with your body and your physiology. And part of that actually helps to function your immune system. There can be harmful bacteria inside of you, but if your GI tract is healthy, it will keep them in check. If your GI tract becomes unhealthy, then opportunistic pathogens can take it over. So specifically, C. diff is a great example of a virus that you have inside, or a bacteria that you have inside of you all the time. But if your GI tract if the microfauna is suppressed, you have a tendency, it'll take over, and that causes severe problems. All right. So the first line of defense in the human immune system is our innate immune system. And these are non-specific short-term defenses. So this consists, one example is physical barriers, skin, epithelial linings, the GI tract, the vaginal linings, all those different stuff. It just physically keeps pathogens out, right? Like you can touch salmonella with your hand and it's not going to make you sick because the salmonella can't transmit through your skin, right? So if you wash your hands and you don't get inside of your mouth, that's not a problem. Mucus layers have a tendency to trap pathogens. So an airborne pathogen, if you breathe it in, if it gets trapped in the mucus just ends up in your GI tract. It ends up getting destroyed. IgA is an abundant antibody found in the human immune system, and it's one of the most abundant antibodies, remember, and it basically binds to all kinds of foreign antigens and tags them for death. And it's not specific; it just finds specific, or it just binds to broadly things that are not you and tags them for death. So it's very non-specific, but it's just non-specific to stuff that isn't you. It wouldn't be very helpful to have IgA floating around that binds to self cells, because that's not how you get an autoimmune disease. Cytokines are also, they're basically cellular messengers that have some kind of immune function. One of the things they do is they actually help regulate a fever state. And fever is kind of an interesting thing wherein the body is saying, okay, we're going to turn up the temperature, and I think my cells will last longer than your cells, right? Because like you can die from a fever. If you have a fever that's too high and enough of your proteins get denatured, it'll cause you to die. But your body is basically making a guess that if your body turns up the temperature, it can better withstand the increased temperature than the bacteria can, which is no necessary guarantee of it. And so I kind of ended up in a rabbit hole reading about if you should treat a fever or if you should not treat a fever, because a mild fever can help speed through the progression of the disease. So if you have a fever of 100 degrees, 101 degrees, a relatively minor fever, your body is not at any risk, right? Like you can stand 101 degree temperature for a couple days and it's not really going to do anything. So by giving Advil, aspirin, Tylenol for 101 degree fever, you're kind of just working against the body's own immune system, right? But if you have a fever that's 103, 104, 105, that can cause serious damage to an individual. So that's definitely one where you should definitely treat the fever, right? So it's kind of this trade-off approach, and this graph kind of shows it. If you are in a situation where the cost of the fever is more than the benefit of the fever, you should treat the fever, right? And you should, that's pyrexia is the name for fever. And that's when you're starting to get in the temperature range of like brain injury, heart failures, septic shock, if your body goes into septic shock. But if it's a non-severe infection, just let the fever run its course. You might actually be able to get over the sickness faster. And so this kind of leads into my hypothesis that if you can tolerate the symptoms of a cold, don't treat the cold. Like the symptoms of the cold are oftentimes your body trying to fight the virus or bacteria or whatever it is. So I try to just let the symptoms run my course as long as I can handle it. Another really interesting thing is that there's something that has to do with macrophages need iron, right? And you have some free-floating iron in your body. But one of the things your body does when it faces an infection is actually take up the iron in your body and get rid of the free-floating iron so that the pathogens don't have access to it. And so like iron deficiency during a fever or an infection might actually help the host overcome the bacteria more quickly. The book also talks about these things called PAMPs, which are pathogen-associated molecular patterns. And they're basically things that are found in a vast variety of microorganisms that tend to infect us, and specifically unicellular microorganisms. So they're things like flagella proteins, cell wall proteins, things that you don't have in your body in high abundance, right? And so if the body sees these, it will recognize it as a foreign body and it will attack it. Okay, so when we're talking about the human immune system, the adaptive immune system is really the big gun out there, right? Like this is what causes, or this is what causes us to have broad immunity to a wide variety of pathogens. It's really the important thing we need to talk about when we're talking about the immune system. And there's two different types of the adaptive immunity. There's cell-mediated and humoral-mediated. Okay. Cell-mediated is a direct attack. So basically, these are the killer T cells, or T cells, right? So they will recognize, cells will present proteins that they're like producing and functioning, And T cells will basically come around and check and see what they're making, right? And so if it comes around and it sees that it's making something that's not a human protein, the T cell will will basically kill it, which will stop it from producing whatever it is, which if it was producing something that's not a human protein, that's a problem, right? The humoral mediated is um, basically these B cells, and B cells secrete antibodies that target um, a specific protein that's been found in the body. And so this is like broad scale immunity, right? So B cells will start to produce just tons and tons and tons of the antibodies. And when they run into the antigen, they will bind to it, and that will mark it for cell death. So if there's any free floating bacteria, if there's any other cells that are producing this antigen that is not human, the free floating antibodies will help identify it. And then that's also what produces our long-term immunity and why vaccinations work, because they produce memory B cells that basically just float around inactivated. But at a certain point in time, if they start to run into that antibody again, or antigen again, they'll start dividing, reproducing, and producing those antigens, which will cause it to respond to it more quickly. We've talked about this before, but the MHC proteins are partially what's helpful. MHC1s basically are found in all cells. And what they do is they basically just say, hey, this is what we're making right now. Like, this is a piece of the protein that we're making right now. So basically it's showing, oh, well, right now I'm producing, human collagen fibers. So, I'm not infected. But all of a sudden, if it starts presenting, oh, I'm producing viral collagen fibers, then that indicates that it is infected. MHC class 2 are only found in antigen-presenting cells, and basically what they'll do is after a cell has been phagocytized by producing, because it's been producing a foreign antigen, it will take that foreign antigen and it'll present it to different parts of the immune system, and will basically say, hey, we found this thing, you might want to be on the lookout for it, which will then activate our immune system. So one of the things your body has to do is it has to produce a variety of different antibodies to foreign antigens, but it doesn't want those antibodies to bind to self-antigens, right? Because if it starts binding to self-antigens, that's how you have autoimmune diseases. And so basically it's the responsibility of the thymus to train T cells that respond to all these different types of antigens, right? And so what basically happens is in the thymus, they're exposed to a wide variety of MHC molecules, right? And if they respond to them, they survive, but if they don't respond to them, they're destroyed, because it really doesn't matter then. They're not going to do anything. So then it exposes it from self-antigens, and if it binds to the self-antigens, right, it's allowed to be destroyed by apoptosis. But if it doesn't recognize the self-antigens, it's allowed to divide and reproduce, and then it becomes a mature T cell. And so in that way, your body basically trains it or eliminates all the T cells that will respond to self-antigens, right? And so it basically has, it produces antibodies that respond to foreign antigens only and not self-antigens. The funny comic. Okay, so autoimmune disorders are basically caused by dysregulation in the immune system. And this is largely caused by mistaking self and not self. Well, it can be caused by three things. The first of which is pretty self-evident, which is confusing self or non-self. So multiple sclerosis is an example wherein, for whatever reason, your immune system targets the myelin sheaths, the Schwann cells that produce myelination. And this causes various different motor and sensory neuron effects. and it can basically cause paralysis and death. Type 1 diabetes is basically your immune system targeting the beta cells in the pancreas that produce insulin, and so then they're no longer able to produce insulin. Because the immune system also causes inflammation. Inflammation in itself can cause a lot of problems. So Crohn's disease and ulcerative colitis are two examples of diseases that are largely autoimmune diseases. And it's basically the human body targeting things in the gastrointestinal tract that usually wouldn't cause problems, but in this situation, the autoimmune is overreactive and it causes inflammation and swelling, which tends to cause gastrointestinal distress, which is not good. Yeah, avoid those. And then you can also have an overactive response. So a really, really rapid response, really, really strong response to something that's relatively benign, like peanuts, and that causes allergic reactions, which is basically your body treating something as a foreign pathogen or a harmful pathogen that is really just like food, like peanuts. One of the interesting things is the risk of autoimmune disease is basically inversely correlated with infectious diseases. So if you have a high risk of infectious disease, then you tend to have a low risk for autoimmune disorders. But if you have a high risk of infectious disease, or if you have a low risk of infectious disease, like in the United States and Western Europe, there tends to be higher rates of autoimmune and inflammatory rates. And people have noticed this and they've speculated about it. And they have a couple different hypotheses, the first of which is known as the old friend hypothesis. So some of the treatments for infectious disease could be eliminating beneficial microorganisms. So for instance, antibiotics themselves could be killing beneficial bacteria in the GI tract, which is then causing autoimmune and inflammation diseases because other foreign pathogens are now more prevalent, or they're not there and they should be. It could also just be, never mind. Friend hypothesis is basically an explanation trying to think about this. And so the idea is the lack of beneficial commensal bacteria in our GI tract causes our immune system to be activated when it shouldn't be activated. And it causes it to target beneficial bacteria in the GI tract, right? So basically, what they found is that if you raise rats in a sterile environment, they develop an abnormal immune system. So some exposure to pathogens is required for your immune system to be trained properly, right? And so if you're never exposed to any kind of pathogen, your immune system could overreact or underreact or inappropriately react, or it could target things that are not necessarily pathogenic and don't really need an immune response, right? Interestingly enough, something called helmet therapy is something that's emerged. And it's basically, they're finding that individuals who are infected with helminths have less autoimmune disorders because the immune system is basically occupied trying to take care of those helminths the entire time. It's really, really interesting. It's really controversial. And I pulled a citation looking at helminth therapy to learn a little bit more about it because it seems so counterintuitive that infecting someone with a disease could actually help them with an autoimmune disease. But there appears to be some evidence to support that. But like I said, it's highly controversial. And it's something that's interesting. Like, has our world become too sterile? And we've previously talked about the gut microbiome and how a more diverse gut microbiome can be beneficial from a nutritional standpoint. So maybe, I don't know. Public health is an evolutionary effect because you can think of it as part of our extended phenotype, right? So like the ability to monitor and control our environment is something that is under evolutionary selection, right? Because individuals who better control their environments and better manage waste and diseases are going to divide and reproduce. So the most famous public health intervention actually involved cholera. And so this doctor known as John Snow basically was aware that there was a severe cholera outbreak in London. And so he basically was able to, through disease tracking, he was able to isolate that most of the cholera was coming from a single pump on Broad Street, right? And he was able to get that well shut down, and when that well was shut down, the cholera outbreak stopped. And it was found that the cholera was coming from inadequate treatment of human feces at the time. And so that really drove the invention of, a dedication to better removal of human waste, shall we say. Funny meme. There's another, the Gambian drought is another one that's really interesting. So in Gambia, they had a situation wherein they had a hunger season and they had a dry season, right? But basically, because they were subsistence farmers, there was high availability, right? And depending on the wet season or the dry season. And people born in the hunger season showed a tenfold increase in disease burden. And this was just that they were born during that period of time, right? And they were developing during that period of time. And they found out that thymus development is sensitive to early life nutrition, right? So if you have less than adequate nutrition during that critical window, your thymus isn't as developed, and so you are more prone to infection because your T cells aren't as effective. And this is likely an evolutionary trade-off, right? Your body doesn't have adequate nutrition. It has to decide where to use it and how to spend it. And so ultimately, by sending less resources to the thymus, you are still developing the neurons and the heart function to hopefully get to a reproductive age, and then you're just traded off later. Oh, and then also they also found that they were less responsive to vaccination. Vaccination is probably the single most important. Oh boy. Vaccinations could be the most important invention of the last 200 years. The polio vaccine, the eradication of polio, the eradication of smallpox was a huge deal. The polio vaccine as well. The polio vaccine was invented by Jonas Salk in the 1950s, and he actually gave it away freely when he could have made billions and billions of dollars. He was still a very wealthy man. Like, he won a Nobel Prize, right? But it's estimated that he saved 2.2 million, or reduced the caseload 2.2 million by 2.2 million people. The smallpox eradication effort is probably one of the single greatest human achievements. We were able to completely remove smallpox. And you can see, I mean, smallpox was basically between 5 and 15% of deaths before the vaccination became common. And then we decided to eradicate it. And nobody has died of smallpox in 50 years. And now there's only hypothetically, there's only 2 vials of smallpox left in the world. Measles. Measles used to be really not a good disease, right? But a vaccine was invented in 1963, which helped reduce it. 1977 or 1971, the MMR vaccine was induced, which is measles, mumps, and rubella. In 1989, the double dose was recommended. I only got a single dose of MMR because I was born in 1985, and so I was treated before that time, so I actually had to get a second dose of it. But large numbers of my generation are actually not immune to measles when they think they are. So I got to this slide and realized that it was not making the point that I wanted it to make. Anyway, so antibiotics is also, I mean, like, you can make an argument that they're just as important as vaccinations, right? And so the graphic right, you can see the introduction of various different types of antibiotics has led to a dramatic reduction in deaths per million individuals. It's also made possible things like surgery. Surgery is a very risky thing if you don't have antibiotics, because the inside of your body is basically sterile, and if bacteria get in there, they have abundant food supply, plenty of warmth, and abundant resources. And so an infection, a post-surgical infection, is very bad, which is why they pump you full of antibiotics following surgery. I would argue, I have argued, and I would argue that antibiotic resistance is probably one of the greatest threats that your generation will face. And we're frankly running out of antibiotics. Um, and this is an evolutionary problem, right? Because what happens is if you expose a bacteria to antibiotics and it kills 99% of the your gut bio, or like 99% of the bacteria, that means 1% survived. And that 1% probably survived for some characteristic that it has. And if that characteristic is genetically heritable, you've supplied incredibly strong evolutionary pressure to an organism that's going to evolve at a really rapid rate. And so that creates that selective pressure creates a very rapid evolution. And so in the course of less than 100 years, we're starting to see our antibiotics become less and less effective. And so much to the point that penicillin is basically useless at this point. There's only a couple bacteria that you can use it for. And we haven't found a new antibiotic since the 1980s. And so if we run out of antibiotics, we're going to have a really bad time, and we're going to have to come up with some solutions. Interesting implication for vaccinations. It's similar to what's going on with antibiotics, but it illustrates that microbes evolve very rapidly, and this can create headaches for researchers. So HIV was a HIV. It's a disease, right? It's a very deadly disease that popped up in the 1980s. And so they're like, okay, we need to find treatments for this. And so they utilize antivirals, one of which is AZT. And AZT is basically thymidine with a slightly different structure. Instead of having a OH at the end, it has an N3 base, right? And so that's important because if you're going through DNA replication, you need that open OH to add another DNA molecule there. But if there's an N3, the DNA polymerase can't add another nucleotide, right? And so it will insert the AZT into the genetic code, and then it'll stop it from happening, which basically stops viral replication. If you stop viral replication, the individual has a chance to fight off or at least delay HIV. The HIV that are in there will stay alive, but it gives your body's immune system a chance to wipe it out. So when they discovered AZT, they're like, hey, we cured AIDS, woohoo! And they were all excited, and so they started to treat it. What they found was that AZT eventually, it would initially very much reduce HIV viral load, but then it would bounce back a little bit later. And so what happened was you would initially wipe out all the HIV that were susceptible to the AZT, but some HIV viruses had DNA polymerase that would not pick up the AZT. And so it wouldn't treat it as a nucleotide. You could tell the difference between the AZT and the regular thymidine. And so that would cause them to not use it, so they were resistant to AZT. So when you apply the selective pressure to the wild type, the normal HIV viruses, the resistant mutations started to proliferate, and then the patient was HIV positive again, which basically means that there's a certain level of HIV found in their white blood cells. What they found works is what's known as combination therapy. And so basically what they do is they will utilize a couple different drugs, a couple different antiretroviral drugs against HIV, and they'll rotate this cocktail. And basically what happens is your body will never be able to fully eliminate HIV, but the rotation of the different diseases or the rotation of the different drugs allows you to keep the viral loads at a level where it doesn't cause health outcomes, which is really important, right? And so at this point in time, HIV, if you live in a Western society with access to modern medicine, is basically a disease that you'll have your entire life, but will likely not kill you as long as you continue to have access to those drugs. But I'd like to emphasize, as long as you continue to have access to those drugs. There's also side effects associated with those drugs. They tend to cause gastrointestinal stress, but it's better than dying. Okay, so we're going to talk about this thing, and it's really, really interesting. So it is a parasite known as Toxoplasma gondii, and so it is a unicellular eukaryotic parasite, right? And it has it has what's known as a rodent-cat dual life stage. A lot of parasites have a dual life stage, wherein they have one host that they reproduce asexually in, and then another host that they reproduce sexually in. I don't know why. Parasites don't make a whole lot of sense to me. But the sexual host is known as a definitive host, right? So it reproduces sexually in cats. So that is the definitive host. It can reproduce in all other organisms, but it tends to reproduce asexually, right? And it can basically infect any warm-blooded animal, right? But its preferred infected individual is rats and mice, okay? And it does this because the parasite is actually able to alter the host behavior. And one of the things it does is it changes the preference for cat urine. So rats and mice that are infected with sufficiently high levels of T. gondii actually prefer, like they seek out cat urine, which ultimately leads to their own death. So the T. gondii are actually altering the behavior of the host, causing them to kill themselves, which is really, really, really interesting. What's also interesting is that this parasitic protozoan can infect humans. And it's also been shown that there is some evidence that the infection of toxic T. gondii in humans will actually alter human behavior as well. Yeah. And in case you're wondering, there are in fact T. gondii infections in the United States. And it's high, not as high as Europe, but it's high and it exists.